Hello ladies and gentlemen, welcome back to C.A.R. Entertainment. I'm your host Alan Hamilton, and today guys we're going to be talking about A22 biology, but in particular we're going to be moving on to a brand new sort of topic. We've done respiration and photosynthesis, now we're doing the genetic code, so DNA sort of. We're going to do a wee bit of DNA, then we're going to actually go into genetics in particular. So we're going to start off and talk about genetic code and transcription, essentially how do we make proteins, and then we'll talk about translation, which is the second part of that. So we'll start off with transcription. So we'll give a wee bit of background on the genetic code to start with. Well, what is a gene? You need to know that a gene is a sequence of nucleotide bases which determine the order of amino acids in a protein which is produced. Genes are a length of DNA that act as a coding device for the synthesis of a protein. So a gene codes for a protein. The main features of a genetic code. Well, a free base code is a codon. Essentially, just know that a codon is a sequence of free nucleotide bases. So if you have ATC, that is a codon, and then obviously they'll have the complementary base pair matches for that. So the genetic code is a degenerate code, as there is more amino acids than one triplet code essentially. So what this means is that there's more triplet codes, or there's more codons essentially, than there are amino acids. So there's more triplet codes. You can get ATC, AGA, free A's, all that there. You go through all of the possible things and you'll get to these 64. But there's only 20 amino acids. So this means that a triplet code will code from, an amino acid can be coded for by multiple triplet codes. So 3 or 4 triplet codes could code for one amino acid. The DNA code is also a universal code, as it codes for the same amino acids in all organisms. So this is what I just said. It's a non-overlapping code, so the triplet codes will be read separately. So if you have A, T, G, C, A, whatever, you'll read that as A, T, A, C, T, G, etc. Along you go. So transcription then, in particular we're talking about messenger RNA to start with here. Well, messenger mRNA, the DNA will be transcribed into single-stranded RNA, which acts as the messenger. So you have DNA with a double helix that will be then transformed into a single-stranded RNA. The reasons for this is that DNA is too long or too large to move out of the nucleus, and you're better to keep the DNA in the nucleus, as it's better protected from the possibility of damaging changes. And therefore, when DNA produces multiple copies of messenger RNA, many proteins will be synthesized simultaneously. So on to the structure of mRNA, just a little bit of background. mRNA is single-stranded. It has a ribose pentose sugar. It has the presence of uracil as a base instead of thymine. So you then have mRNA is also the facility for transporting DNA code from the nucleus to the cytoplasm. It is only complementary to a relatively short section of DNA, and it is much shorter than DNA as well. So this is a comparison between DNA and RNA. Well, the size, obviously DNA is longer than RNA, RNA is shorter. Nucleotide arrangement, RNA has a single-stranded and DNA is double helix. Sugars, the RNA has ribose, DNA has a deoxyribose. Bases A, T, G, and C, always known as adenine, thymine, guanine, and cytosine for DNA, and then in RNA it is adenine, uracil, guanine, and cytosine. The location of DNA is in the nucleus, and RNA is produced in the nucleus, but you can find it throughout the cell. So transcription then, let's go through it. Well, at the start of transcription, you'll have an enzyme DNA helicase which will separate two DNA strands by breaking the hydrogen bonds, and this will cause the double helix to unzip. Then RNA polymerase will bind to the region of DNA near the beginning of the gene, which is going to be transcribed. The first exposed strand, the export strand, will be used as a template for mRNA production. As RNA polymerase will move along the mRNA strand, 3 nucleotides will be matched up by complementary base pairs, and this will form hydrogen bonds. The RNA polymerase will bind the newly arrived nucleotides. As RNA polymerase moves along, the DNA double helix will reform, and the enzyme will eventually just stop codon in the genetic code, and it will stop. The DNA polymerase will stop, and therefore it will release a fully formed mRNA, which will be produced complementary to the nucleoside base sequence on the template strand of DNA. So you've unzipped, you've got 2 strands. RNA polymerase will move along one of the strands, and it will bind nucleotides to it, and therefore that mRNA strand will be released. Thanks for watching this video. I've been Alan Hamilton from C.A.R. Entertainment, and I will see you next time.